So, thank you everybody for being so soon here after lunchtime. So let's start with the second session of the CAMDA. Now this short session, we will have 4 talks. 2 of them still on the challenge of the microbial challenge, and we will start with 2 more on the daily challenge. So our first talk is going to be given by Chih-yu Chen from the Public Health Agency of Canada, and its title is Systematic Evaluation of Microbial Abundance from Metagenomic Sequencing Data for Machine Learning Prediction of Sample Origin. Thank you.
Thank you. Hi, my name is Julie. I'd like to first thank the organizers for the generous time to present my research for the CAMDA challenge, and also the 5 reviewers to provide the feedback on the extended abstract. So a little bit of a forethought on the reason that I participate. I work in the National Microbiology Laboratory within the Bioinformatics Core Facility. So usually we have the genomic core data sets and proteomics core data sets that comes through us, and I work as a data scientist over there. So this is in the federal government of Canada. I don't generally do the heavy research, but usually it's providing resources and all that. But previously, I've analyzed 16S gene sequencing amplicon sequencing data set using random forest classification. I classified immune-related diseases versus healthy control samples. I just thought it was a good opportunity to participate in the CAMDA challenge with the metagenomic data and with application of machine learning. The key is to participate, contribute, connect with you, and refine my practice at work. So just to give you guys a little bit of sense for along with the speakers in the morning as well, we only had roughly 4 months to work on this, and I am going to present to you the journey within the 4 months. So this is really very much work in progress. So we were provided with quite a bit of data sets. The ones that I used in the talk today are listed up here. It included 16S data from Earth microbiome, the Boston pilot data with 16S and whole genome sequencing, and also the main challenge data with the shotgun metagenomic sequencing and the mystery data set. So what it is generally we extract the feature abundance. The 16S OTU table was provided, whereas from shotgun metagenomic sequencing data, we extracted the taxonomy abundance using Kraken2 and Bracken combo. So this was done by my colleague, and she used a custom in-house database for it from RefSeq. Additionally, I normalized the data sets using cumulative sum scaling, using metagenome-seq package with 50th percentile as the default setting. And additionally, there were one filter, which is at least one read in more than 8 samples to keep this particular either species, if it's the species table, or genus for genus table. All the data sets has been logged to transformed before the machine learning step. Along the analysis, I've used R, Bioconductor, and other packages to power the analysis. So there are 3 main questions that I tried to address here. The first one is, how do 16S datasets versus metagenomic sequencing dataset compare? That's using the Boston pilot dataset. The second thing was more building the machine learning model and see how it compares with cross-validation, etc. Lastly, I wanted to ask how is the prediction? And you've heard from the morning talks, you probably know what to expect, sort of. I will be providing the numbers by the very end. So oftentimes we have the truth, the ground truth, and whatever data sets that gives us, right? So here I'm essentially comparing, also re-evaluating the differences between 16S-based data versus shotgun metagenomic sequencing data using the pilot. I know that there are already literature on it. It's already been known there are some known characteristics in either pros and cons for either of the data set, but I found a little bit of discrepancy in terms of one paper saying that shotgun data had increased detection of diversity, while the other mentioned that the amplicon sequence revealed 27% more families. So I was wondering what's going on over there. What I did was that I compared the number of taxa count obtained from each of the data set. So here you're looking at Kraken and Bracken combo, where because it's metagenomic sequencing, you obtain more species, more taxa other than bacteria. So I added this column right here where you can see the counts are a lot higher than 16S. Just one note here is that the counts here is if there is presence detection in any of the Boston samples. So even though we see 3,000 here, it doesn't mean that all Boston samples have abundance in these all 3,000 species. And you can see that there's overlap right here. But if you look at MetaFlan table that was provided to us from the challenge, what you're seeing here is much lower number of counts right here. And in fact, it's higher in species, but there's no overlap here. Okay, and the next thing is what I observed is that you do see above species, all these rankings have higher number of counts in 16S, compared to shotgun sequencing data. So that's using the MetaFlan table, whereas it's very different when using Kraken and Bracken. So that's one thing to keep in mind of where the discrepancy that we're seeing in the literature. There are differences between analytical approaches and databases. From what I talk to others, oftentimes people refer to the differences in databases. We haven't had the time to use the program and use the custom database to do the comparison, but it's one of those or both of those factors. Now, I was curious to see whether the log2 abundance between these two technologies actually have some sort of agreement, because the thought is if there is some sort of pattern that we can capture, perhaps I could utilize the EMP, the Earth Microbiome 16S data sets to sort of help us do the prediction later on. So what you're seeing here is the x-axis is the KNB log2 abundance, and y is the 16S abundance. Each of the scatter plot represent a taxonomic scale from species all the way to order. And for each dot within the scatter plot, it means a species within a sample. And I overlaid all the samples on there. What you're seeing here first is that there is inflated zero in the 16S data set. What you're seeing right here with the histogram showing the distribution. Secondly, you see greater variation at lower abundance level and higher agreement at higher abundance level. Thirdly, if you look at the correlation, it gets higher as you go up the taxonomic ranking, and obviously it must be related with more abundance as well. Now, if you want to look at MetaFlan to do the same thing, first thing I want to point out is that the correlation is slightly lower in MetaFlan, where there is a correlation. So there's no overlap in the species. And so another thing to point out is that now the inflated zero is actually in the shotgun data from the MetaFlan table. The 0.2 and 3 that I mentioned previously still holds. But that just goes to show that, again, the database, also the approach really matters quite a bit. So at this point, I wanted to have a quick view to see whether I'm comfortable incorporating both data sets, both technological data sets. Here, what we're seeing is with Bray-Curtis dissimilarity from all samples, the technological data sets actually cluster together. So purple represents the shotgun data set, and orange is the 16S. This is also reflected in the first component of PICO-A plot, where you see the separation of the purple and yellow. And what you can see a little barely, is that there are some pattern for the biological surface that the samples were collected from at the third and fifth component. So my conclusion here was that, okay, there is intrinsic difference between the two data sets, even if it's on the same sample, and it's overpowering biological pattern. So we have to treat it before we can use it. However, I ran out of time to work on this. I had to quickly jump into the challenge. But to keep in mind is that when we get to higher abundance, there's more higher correlation. So now moving to the step for the challenge, because of the knowledge that I got from the previous section, I decided not to incorporate the 16S, at least for the time being. We've seen Carlos' presentation of how he incorporated it. I didn't go in that direction. So essentially what I did was to do supervised machine learning on the data set separately and also compare it between the taxonomic ranking. So here's the data sets from the cities in the training data set. There were 16 cities aside from the Boston pilot. One of it, which is Sacramento, had single end data. The rest had paired end data. And also, I'd like to draw to your attention that you see a higher density in Europe. And below the equator, there are actually 3 cities. So I did the usual processing of the data sets and look at it with Bray-Curtis PICO-A plot. What you see here, I'll just quickly touch on it, are the 2 cities from New Zealand that are clustering quite separately from others. So there are cities that are more clustered together, but there are some overlaps between cities. And you can sort of get a sense of that in the heat map on the right as well. But keep in mind, this is 2-dimension that we're looking at only. So here comes the challenge that you've heard all over this morning, where we have mystery samples that were not previously trained before. Right, so that influenced how I tackled the challenge quite a bit. If we were to look into the existing supervised machine learning approaches, you could sort of make them into 2 categories, classification and regression. And if they think of the challenge, oftentimes people would go with classification, which includes all these previous work that uses random forest, SVM, Bayesian approaches. One of the key limitations is that one can only predict cities that were trained on. So I opt to use the regression approach. I hardly see existing machine learning approaches to predict the coordinates. There are a little bit of work that's statistically inclined or using correlation to look at the latitude. These are the few studies that look at the latitude. One of it was the distance from equator, so they took a different route. So the key thing to keep in mind is that with the assumption of the regression, like if I were to choose linear regression, I'm assuming there's a linear trend along coordinates. So quickly, the source of prediction approach that I've taken was multivariate regression with lasso regularization. With lasso, it's to avoid overfitting. And multivariate is when I model both latitude and longitude together. So again, the feature data, if it's a species model, it's species abundant table. And so that's essentially a simple linear regression model. And again, for those who are not aware, the lasso regularization essentially infused a penalty in there where we are minimizing the loss. And it has a lambda factor, which I chose to be one standard error away from the lambda with minimal loss. And here the loss is computed as mean squared error. And you're comparing between predicted value minus the true value, and take the square and take the mean. Because we have 2 variate, longitude and latitude, so it's essentially summing both of the MSE up. And as a comparison, I also did, by the very end, which I'll show you, classification using multinomial logistic regression and also with lasso regularization. Okay. Now, if we just have a look at the numbers, this is the model performance. It's very similar to what I submitted before. I just want to give you a heads up. In my extended abstract, I actually only used paired end data sets for training, which you'll see in the next slide. But afterwards, I incorporated the Sacramento data, which is single end, because I originally wasn't comfortable incorporating data sets from different techniques. And here, this is models done separately for 16S RNA. The family model performed better than others. And from the MSE, also, I looked at the R squared for latitude and longitude. Now, for the shotgun data set, species performed the best. It has better performance compared to others. And here's the result. It was a little unclear when I see the MSE. I was like, what is that? I mean, I do know what it is, but if you want to visualize it, what I did was in the extended abstract, I provided this plot, which is the truth versus prediction on the y-axis. And you're looking at longitude on top, latitude on the bottom. My first reaction is, okay, there's a lot of residual is pretty high. And so this was the family model for EMP, which is the best model compared to the other two ranking. And over here, it's the species model with the paired end data that I had. So this is tenfold cross-validation result. Now, moving on to later on, because I left out the Sacramento single end data, so which is in red right here, you can see that the latitude was predicted quite nicely. Yes, there is still residual, but the longitude was really horribly off. And lastly, with the other black dots, those are paired end data treated as singles. So what my colleague did was she essentially ran it as singles, so you'll have two data sets per paired data. And so that sort of got me a little comfortable thinking that, okay, maybe single end data is not that far from the paired end. That's why after the extended abstract, I incorporated the Sacramento data into the analysis. And you may be wondering why I chose to do the bivariate, which is the multivariate approach. So here's a comparison to univariate, which is if I were to model longitude and latitude separately. Right here, you can see the MSE is higher. This is essentially the sum of the two in the model, and you can see that bivariate is predicting like with lower MSE. So now coming back to the multivariate model, I further did nested tenfold cross-validation to be sure. And in the very, very last sentence of my extended abstract, which landed me the talk here, I said, further testing using leaf one city out in a nested loop would be more robust for a model evaluation. And that is because we're predicting an unknown city that we haven't sampled before. Because when we do cross-validation tenfold, we're leaving some 1/10 of the samples out, and chances are you do already have samples from the same city, right? So this is for addressing the challenge of predicting new cities. And so if you just take a breath, and we'll see truth and nothing but the truth. And so this is the result when I leave a city out. I thought maybe I made a mistake, but if you look at Sacramento, it's right here. It's the same one, and it's predicting right here. So that's when you don't have a sample from that city, here's the result that you get. So a lot of people were asking about numbers in the previous talks. You'll get more and more of these numbers. And so by this time, it's pretty demoralizing, really. But it just goes to show that the challenge of predicting a city where no samples have been collected previously, and at least from the approach that I took. Now, I went in to look into, so what are the cities that had higher squared error than others? If you separate them out to longitude and latitude, I have this panel looking this order throughout the talk. So Tokyo and Sacramento had really wrong prediction in terms of longitude, and Sao Paulo has a terrible latitude prediction. And one thing the reason that I put this up is that at the time I was observing what are the cities that performed terribly, and you can sort of see Sacramento's right here, and there are others. There's Tokyo, there's Hong Kong, and right here. So usually these cities are separate from others. Like you don't have a cluster of some neighboring city. So I was hoping that maybe it's because of that, but it's not really, like it requires proof, like you do need to do more analysis on that. So when I talked to my colleagues about these results, my predictions, just so crappy, and so two of them had suggestions. What about, maybe it was too ambitious, you're looking at the entire world, what about if you localize to a continent? And there's more data sets in Europe, so I went and did it, and yeah, so here's the result again. So we'll see a few of these. And so then next, I wonder whether, maybe it's the linearity. I have this assumption. What if I do random forest? At least it's ensemble trees, right? And so here you see the cross-validation. In fact, the R-squared for latitude is a little higher. But if you leave one city out, it's still the same. It's just, yeah. So I wish I have a better news. So then by this time, I decided I'll look into classification. The whole point of using regular regression is that I was hoping that I could predict the cities that haven't sampled before, right? It failed miserably, so I went back to the good old classification and just wanted to see for my sanity how it performs. Again, coming back to the tenfold cross-validation, like we've seen this type of plot from the previous speakers, and obviously with the cross-validation, it looks fairly nice. The only thing that was confusing in the model was between the two New Zealand cities, and there were a few wrongly predicted samples. But if you look at leave one city out, so I was scratching my head how to visualize this. So I ordered the continent here and made the blocks for continent. What you can see here is New Zealand cities nicely predicted to be one another. What about others? So London technically did not do so bad, and Berlin either. At least it's in Europe. And if you continue on, Marseille, and the worst one were the Stockholm, and PXO had half of it predicted to be in Europe and half outside. And so, yeah, we can interpret it for hours, but it's not so good. So at this point, there is still a challenge, right? So I'm glad that I did the leave one city out. It gave me a preparation to know that I don't think it's going to do so good. So I knew this before. So in fact, I was a little deflated. I have work to do. I don't usually do strictly research at work, so I sort of let it go a little bit. So just to complete the challenge, let's look at the result. So again, it's still longitude on the top, latitude on the bottom. And so this is with Sacramento and species model with the regression. And this is what the result is. So it's, for some reason, this was shifted away from the diagonal line. And, well, this one, even though the R-square is 0.42, the slope is quite far from the diagonal. But by accident, I predicted the mystery sample using the model before I submitted the extended abstract because I already had the script there. And this is what you see. It's actually much better. So I messed up along the way. I'm sorry that I don't have enough time to show you like the result throughout for leave one city using this model, but the correlation at least is a little more comforting, except for this one, which is really poorly predicted. And here there is a difference in slope as usual. So then I looked into the squared error of the samples by city, and this is again with Sacramento data, which is the single end included. The Brisbane was really predicted poorly, even though there's New Zealand nearby. And you can see Oslo is predicted poorly as well. And here Chile, in fact, the longitude was okay, well, relatively okay, but latitude was predicted poorly. So I really couldn't see a lot of patterns. Remember I said maybe because you don't have a nearby city, it may have affected. I would think it requires really more rigorous analysis. I can't conclude that observation, whether that's true or not. Okay, so next, the results from without Sacramento. What you see here is that Oslo is no longer predicted to be so wrong in terms of longitude. And if you pay attention to the scale of the plot, this is 40,000, 5,000, and this is 20,000, 4,000. So because of that shift, remember the plot? So originally, if I didn't use the Sacramento data into the training data set, then this would have been the result that I'd be showing you. Now, again, so since I did the classification, let's just, for the sake of it, compare it. This is the result for the mystery samples, and I again boxed the same continent. For example, Vienna was nicely predicted in Marseille, and there are a few things that are outside, some are inside. And here's the squared error plot. I won't go too much into it, but essentially, I realized that there should be some better way to summarize it, which is comparing the MSEs between different models. Okay, so remember I thought I'd try regression, but the result is not really that much better than using classification, even if you don't have the original city. So remember, when you classify without the city before, then what you did is supposedly, hopefully, you predict the nearest one, right? But anyways, the distance, the MSE is actually a little better than regression. But if I had sticked with the one without the single end data in there, that's the MSE. So that may be the direction that I look a little more into, but it has to be some sort of reason why it's performing that way. Okay, so here's the summary. How am I with the time? I hope it's okay. Okay, thanks. So in summary, we've detected differences in sensitivity, and there's higher variation in lower abundant taxa. And so this was for the Boston pilot data, and it also showed that different data analytical tool and database can drastically affect the result, at least the count of your species. And the second point was that when I did the machine learning analysis for both of these data sets, we found higher predictive power at the species level for shotgun data, and at family level for 16S data. That's through the tenfold cross-validation. And again, with the cross-validation result, it showed potential when you have prior training samples in the city. But I want to raise a caution here, which is it's just in the back of my head that if you do different protocols in different cities, I cannot guarantee that one is predicting the real barcode of the city and not the protocol. Then the other point is that leave one city out really highlights the challenge of predicting city without training samples, and this is the case for both supervised regression and classification approaches. Lastly, further investigation really is required to see why the model performed that way when I incorporated Sacramento single-end data. I mean, I could hypothesize it's really far out. It's solo, out in one geological location, but it really requires more work to really conclude that. So there are a few limitations of the study and also about the data too. Keep in mind that we don't have any metadata in this case, and even from the Boston data, like if you look at the surfaces that are collected, I think at some point you can find that there are differences, and there's climate differences. I even looked up MetaSub to see when the samples were collected, because if it were in the southern hemisphere, it's winter and summer, right? It's different seasons. And if you look at the global atlas data, they actually predicted several models for humidity, like a lot of different environmental things. So if you work more with biological data, you know that these are all the hidden factors in there. And if you happen to have like more humid city, then you may be having that signature of humid city, right? So it's really hard to really state the features. So I really avoided talking about top feature here. One thing though, like if you have classification models, top feature is more clear cut because you can just plot the box plot. But if you have a regression model, it can get a little messy. Okay, back to the limitation for the data analytics. I used the longitude and latitude, and remember it's bounded. And with latitude, 190 is actually the other way, right? It goes to the other side. So I actually tried a different approach for that, but we just won't go into there. That's one of the limitations. And other things is to keep in mind of the protocol, batches, and also environment. And also, I mean, this is not something we can necessarily control, but there are different distribution of the training city around the globe. And lastly, because it's metagenomic data, you can actually extract different features, and we've seen that in the morning with k-mers, etc., and also pathways. So I did try a little bit on the functional pathway, but I didn't have time to finish it. Lastly, I would actually do a little more on the quality and filtering and normalization approaches to really compare. I think it's very vital, but within the given amount of time, I had to have something to submit. And so I'd really like to thank my collaborator, Dr. Andrea Tyler. She processed the metagenomic sequencing data because I tried MetaFlan 2 at first with just the typical database. It wouldn't have that much resolution if she hadn't participated. And I'd really like to thank my boss who let me do this, because this is technically not work, but we do have metagenomic data sets, 16S data sets. So all the knowledge extracted from here and from talking to you, listening to your talks, it's very helpful. And also, I'd like to thank my colleagues for the feedback, CAMDA organizers, and lastly, this is where I submitted my extended abstract. I got fed up with working at home on weekends, and so I went to a campground, and that's where it was submitted. Thank you very much.
So thank you very much. Is there any question?
Did you try just getting rid of the relative abundance information and just using the tax as binary, present and absent?
No, I didn't. Yeah, I thought it would be a way to address it and actually denoise it. And I want to comment on one thing too. I also plotted Spearman rank correlation, and when I do that, the plot looks a lot cleaner in terms of separating, because in the morning talk, we have one speaker who actually sort of disbins the abundance. And so somehow it gave me, I can show you this. Oh, here it is on the bottom. What you'll see here is that if it's by ranking, if you look at the Spearman correlation, you see a lot cleaner than Breakhurd is. So maybe there's like even binarizing it or I don't know, using ranking or some sort may help it. I don't know.
I was just wondering the city that was the outlier, that was the single end reads, right?
Yes.
And then you combined it by treating the paired end reads as single end reads. So you took both reads. Perhaps that overweights these cities in a way that it shouldn't if you just took the first read, perhaps?
Oh, so when I took the paired end data, that's from running Kraken and Bracken as paired end. right? So when I said I separated that was when I trained on paired and predicted on singles. Yeah, so that's why it was a lot easier at first to build a model that are paired in, because it's all processed together as pairs, and singles we had a different folder all in singles. Yeah.
Thank you for your nice presentation. In fact, in terms of the Sacramento, it might be also the other explanation, not only the single-end pattern, but Sacramento was also a pilot study where a different approach for swapping and extraction was used. So it probably might explain, because our hope was that the biological signal will be dominating the other factors, but it's clearly seen that when you compare 16S whole genome, you see also that the material and other factors are really affecting this, and we need to find where really the biological signal is. Yeah. So. Yeah. I think for next year challenge, we are just going to provide very, very detailed meta-information so you can also test these additional factors, how they are affecting the classification and prediction.
Yeah, thank you very much for clarifying that. If I may add one more comment, because I knew in the beginning that these were treated differently because I somehow encountered a single, and just from that perspective, if one were to do protocols that are different, either it's best to be consistent around, because I've analyzed ENCODE consortium data sets where they have a specific protocol. So that's one thing. Another thing is to compare, maybe do a pilot where you have this, everybody runs the same sample. That way it sort of helps inform, but I know it's more work, but just to trust the data. I was analyzing this throughout and very skeptical about my model. Okay, so that's all I have. Any other questions?
So if there are no more questions, then thank you very much again.